Today, it's my pleasure to talk with Tomas Horvath. He's professor and chair of the Department of Comparative Medicine and professor of neurobiology at Yale University School of Medicine. He's also director of the Yale Program for Integrative Cell Signaling in the Neurobiology of Metabolism. He's an expert on the control of energy metabolism, how your brain controls energy metabolism in your body and vice versa, these signals going from the brain to the body and the body to the brain that, hunger, satiety. And he's going to talk about what's going on in the hypothalamus to regulate our hunger and satiety. And of course, this is something I've been interested from the standpoint of intermittent fasting and its effects. So welcome, Tomas. You grew up in Hungary, is that right? And I should also mention you're a veterinarian. You got a veterinary medicine degree, and that's, I assume, a lot to do with why you're head of the comparative medicine section, which most places essentially, all the animal research going on, which is, a lot of mice and rats mainly. So welcome, Tomas.
Thank you very much, Mark, for having me. It's been always a pleasure to interact with you, either verbally, in person, or in writing. And I'm delighted to tell you about, basically, if you want to put it that way, my life story, starting from finishing vet school back in Hungary. My father was a veterinarian. His father was a veterinarian. My mother was a dentist. His father was a pulmonologist. And the two of them worked in the 1940s and 50s, one veterinarian, one physician on TB, because the TB was predominantly coming from the cows, and they worked together. So I grew up in an environment where a physician and a veterinarian really worked together. And for me then to jump into medical school was very easy and an easy step. Specifically, to be very honest with you, when I finished vet school back in Budapest, I realized that I really don't have the good skills to become a veterinarian. And I would rather be focusing on research, animal-based research. And I ended up in an OBGYN department, which had a group that was very specifically interested in the hypothalamus. And actually, there was a Hungarian guy who recruited me there, who came there in the 80s. The Hungarians were famous at that point, and still, I think, have some respect for electron microscopy. And the chair of the department of OBGYN at that point was Fred Naftalin, who happened to be also very much interested in the hypothalamus from the perspective of reproductive neuroendocrinology and aromatization and sexual differentiation and so forth. So I was brought in as a postdoc to do a lot of electron microscopy. Basically, that's, I could say, if I want to be honest, that's my only skill set, and that is to do electron microscopy with my two hands, which I did for a long time.
Tomas, so your father was a large animal veterinarian.
Yes. And what's his father?
Oh, wow. I grew up on a farm in Minnesota. My father was a district attorney, actually, but my grandfather, my dad's dad, and him and me and my brother trained and raced harness horses, trotters and pacers. They pull a cart. And so actually I wanted to be a veterinarian, and I didn't get into veterinary medicine school. I really didn't like just memorizing things. And I was, I would get kind of bored very easily. And the way all the courses were taught back then, it's just, they give you something and you memorize it, and you don't necessarily even have to know how everything fits together in the bigger picture.
Absolutely. And one of the worst part was in the first 2 years of anatomy. Because in anatomy, you really had to memorize exactly where the muscle is starting, how it goes through, where it ends. And on top of that, compared to medical school, you had to know the specific differences between different species.
Yeah.
So that really was horrible. I would say it was horrible. Now, it's very interesting. In the same time, retrospect, if I think about medicine or the education of medicine, that's the only objective discipline, because the muscle is there, it starts there, goes there. All the rest, we are constantly debating biochemistry, physiology. It's always a debate. Anatomy is the basis of medicine. It's a kind of interesting background, but definitely turned me off too. Except, back when I went to school, and in Europe, you have many places, including Hungary and other European countries, that you don't have the college system. So from high school, you jump directly to a professional school.
Okay.
But you don't have this 4 years of trying to figure yourself out and make a decision that perhaps best fits you. So I ended up vet school, which was also very difficult to get in. There's only one vet school in Hungary. So it was more like an intellectual challenge, to be honest with you.
Yeah.
Than the devotion to the discipline at that point.
Okay, so you learned about the hypothalamus and its role at the time in controlling reproduction, and you start looking
first project. So I arrived on a Friday, and Monday when I walk in the lab, the Hungarian guy gives me a material, electron microscopy material, said, work it up into a paper. I knew nothing. I hardly spoke English. There were no computers. You wrote everything by hand. And actually, this was my first work, which was to study the relationship between, now we know, MPY AGRP neurons and the POMC neurons, the pro-opioid melanocortin producing neurons. And that was, again, from the perspective of how these two subsystems mediate the effect of estrogen on gonadotropin releasing hormone neurons. But because I had a collaboration with Satya Kalra, whom I'm assume you knew back then, who was the first to show that MPY is a powerful orexigenic agent. In the paper, we hypothesized back in 1992, by the time this paper got published, that the interplay between these two subsets of neurons might play a role in the control of appetite.
Can you back up just a little bit and describe the hypothalamus and why it's interesting compared to other brain regions? It took a long time to figure out the connections, because it's, for example, the hippocampus, there's layers of neurons that seem to be connected in a logical manner.
In a predictable fashion, and potentially a predictable output of that system. And that you don't have in the hypothalamus. And that was, in a way, if I can refer to my experience with neuroscience, the hypothalamus was always the orphan child of brain research. Nobody hardcore neuroscientists wanted to work with the hypothalamus because of that very thing you just described. It's really the cortex, the hippocampus, that's these sophisticated entities that are making whom we are, and this messy thing in the bottom, it's not something that they're interested in. So the interest on the hypothalamus was predominantly from endocrinologists, because they did get an understanding that there are signals coming in and coming from the hypothalamus that are fundamentally important for reproduction, for the thyroid axis, for the stress axis, for growth, and so forth. So it was and still is, although it's changing, dramatically changing, but it still is a sort of orphan region of the body, because endocrinologists approached it predominantly as if it were an ovary, so not appreciating entirely what neuronal and glial contribution are to the functionality of the enterprise. And of course, the people who were working in the cortex and hippocampus couldn't care less about what's going on in this mess, because the tools that emerged over the decades were not really lending themselves to a proper interrogation of this part of the brain. But I was intrigued by the very fact that a single neuron, although right now we associate, let's say, the AGRP neurons with hunger, the POMC neurons with satiety, the reality of the matter is that they are actually involved with all of these many homeostatic functions. And also, homeostatic functions will be impacting tremendously higher brain functions. So that sort of a relationship between this mass and the orderly output of the brain always intrigued me.
So why don't you start with, okay, so say I've eaten a meal, whatever, 10, 12 hours ago, and the food has gone through my stomach. Then what happens that gives me the perception I'm hungry?
Based on what we learned, we believe that the signal comes from the gut through many ways, but the one that I'm going to tell you about is through the blood, which then contains elevating amounts of certain hormones that promote these neurons that promote hunger, such as ghrelin.
Yeah.
Ghrelin reaches the hypothalamus, activates these AGRP neurons. And there is an important consideration that needs to be kept, and that is that the way the circuitry is organized, which was actually in our first paper, is that the hunger eventually turned out to be hunger-promoting neurons are the dominant component of the circuitry. So we are wired to be hungry by default. And that is sensical. It's sensical because if it was the other way around, we would die after birth if we wouldn't have the drive. And eventually becomes a burden when you have an environment where, if you want to call it obesogenic environment, where you don't need that continuous drive. Nevertheless, these hormones signal to these cells, and then these cells suppress those neurons that otherwise would promote satiety. Once you have a meal, then these cells get turned off, and then those neurons that were turned down by the system are activated, and then you promote satiety, which means many different things. It means behavior. In both of them, means behavior adaptation as well as adaptation of the peripheral system to deal with the either incoming nutrients or deal with the situation when you have no incoming nutrients. You always have to find a way to provide for the rest of the body, even if there is not incoming fuel. And when you have incoming fuel, you still have to provide for the body, but you also have to find a way to store the incoming fuel. You don't want to burn all of the incoming fuel.
I've noticed, right, so people that fast for a long time, they'll initially experience hunger maybe the first day or so, and then, their feelings of hunger will decline. And I know that people with obesity often seem to be hungry a lot, even when another person wouldn't necessarily be hungry, having not eaten that time. So is it known what, and then we have a cat, right? And we normally feed it twice a day, but then lately, my wife and me sometimes have been giving it treats in between. And now it wants a treat all the time, because it's rewarding. So I think I guess I'm asking kind of a complex question, but what's kind of the temporal regulation of these processes? And
so I think
yeah,
the first issue that you mentioned, that if you fast for a prolonged period of time, that the initial hunger diminishes.
Right.
I think this is a very interesting phenomenology that we know very little about, actually. Yeah. And there is a second component to that, actually. I experienced it, I'm sure you have, and I've seen that on my dog as well. So I give him the food, and then she's sort of, not really interested in it, although she hasn't eaten since the morning, or she hasn't eaten most of the day for that matter. And then once she starts to eat, then actually the hunger kicks in.
Yeah.
Then actually you develop hunger and start to be eating as much as in that moment you will take in, which obviously has many personality trait issues. And
that makes sense, Tomas, right, from an evolutionary perspective, because if you have like one big source of food, it's probably good to eat as much as you can, because you may not, it may be a long time before there's another.
Exactly. That's why I always have a little bit of an issue when somebody, especially functional imaging people who I had a good chance to interact with, a great scientist, that they show you these two pictures, a chocolate cake versus a six-pack muscle. And how you're supposed to be making prefrontal cortical decisions about how much and what to, or salad and the thing, in order to have six-pack. But actually, the intent, and I'm might sound a little bit off here, but the real intelligent brain wants to eat chocolate cake because you don't know what's going to happen tomorrow, and you don't know what's going to happen the day after tomorrow. So to build food, energy into storage is a very intelligent way of having the body work. Of course, when you constantly push that, it's not a great thing to have, but it's per se that you are driven to overeat. I think it's sensical, again, from an evolutionary perspective.
Yeah. Okay, so when you haven't eaten for a long time, ghrelin is released from cells in the stomach and goes into the blood.
Sugar levels go down, glucose goes down.
Yeah.
Fatty acids are released from the storage because fatty acids become then
ketones,
an important fuel for the rest of the body. And the constellation of these events are important for these neurons to get activated, and eventually to these neurons to get to the point where they will trigger not only hunger per se as a perception, but also all sorts of behaviors that will make you hopefully successful in the environment to find your food. And that means exploration. So that's the other thing I frequently argue, that there is no human behavior that you could not connect or should not connect to hunger and satiety, and of course the between states.
Okay, so then these AGRP neurons are activated by ghrelin, and then somehow they have to be connected to other brain regions that are involved in decision making or the perception of hunger.
So the way we actually envision it is that it's not a single pathway as many people would hope to have, that A, B, C, D, and there you have a cortical output, or A, B, C, D, and then there you have the autonomic output. But actually it's a diffuse way of communication from these hundreds of neurons to many parts of the brain. We studied the ascending dopamine system, which plays a role in these things. And this ascending dopamine system feeds into your reward components in the ventral striatum, but it also feeds into the prefrontal cortex and other cortical regions. So there is this neuronal, still diffuse way of communicating up to the whole brain, because we look at the brain as an organ, and as much as I was not intrigued at the beginning, 30 some years ago when I started, by this view of the brain as any other organ by the endocrinologist, eventually I sort of came around and say that the brain is a synchronized, it's an organ that has tremendous synchrony, and that synchrony is tremendously impacted and regulated by these bottom-up communications in response to food or not availability of food.
Okay, so then when you eat a meal, what happens that essentially tells you to stop, you're full, or to to tamp down your hunger after you've eaten?
I do believe that glucose plays an important role in that, the elevating amount of glucose. When it's combined with the lipids, I think that is an interesting response, and of course amino acids and proteins. And then you start to have these neurons get turned off, and the neurons that are promoting satiety get turned on. And the downstream effect will be altered allocation of fuels, storing in the fat, putting in the muscle, and realign your entire behavior profile.
So when then leptin's released from fat cells into the blood, it goes to travels up to the hypothalamus. And now, is it acting only on the POMC neurons, the POMC neurons?
No, actually, no. It's acting on the AGRP neurons as well. Oh. And if you look at the distribution pattern of leptin receptor, they're much more widespread than just these two subsets of neurons in the hypothalamus. And one critical element in this communication between the body and the brain will be the autonomic nervous system. So vagal afferents will signal up through the NTS and the parabrachial nucleus to participate in this satiety, accomplishment of satiety, which has been somewhat overlooked in the last 30 years because of the discovery of the leptin and eventually ghrelin. And there was, and there still is, some level of emphasis put on the humoral component of this communication versus there is the neural component of the communication.
I had I don't know if you've met Kevin Tracy.
No.
Yes, he's done a lot of work on these vagal afferents, and mainly related to what he called, this was his work, that kind of an inflammatory reflex thing, whereby the vagus is affecting inflammation, but these afferents coming from sites of inflammation apparently are sending signals up,
like the GDNF15, that kind of
yeah.
Well, that's for me, it's an interesting argument. I was at a meeting in Korea, and there was one of our colleagues who distinguishes between healthy satiety versus sickness-related satiety. For me, it's difficult to distinguish between those two. Even if you think about, if you eat a large meal, you can be nauseated, and this nausea, or if you look at these medications that are now very successful, they have a very strong nausea promoting action. So I'm not sure whether that nausea sickness is not part of this continuum. One extreme is the starvation versus the other extreme is nausea or cachexia, if you want to put it that way. I consider them as a continuum, but it's my personal view of the system.
It's kind of a 30,000 feet level. The idea is that when you're sick, it may not be good to eat because you need to devote
correct
resources not to digesting food but to fighting the infection or whatever.
And one of the intriguing things for me right now in this debate, which I'm writing a small little thing on, is if you take these three things: calorie restriction, where you self-limit your food intake; drug-induced suppression of food intake; versus cachexia, where you have diminishment of food intake. And I think one of the intriguing questions is it the decrease of food intake that is benefiting you, or the associated adaptations of your body to how you allocate fuels, how you change your hormonal levels? And it's something that I'm very much interested in pursuing.
Yeah.
Looking at the cachexia example, it doesn't seem to me that artificially suppressing appetite when you are not involved in that suppression is beneficial, but again, we'll see how it goes.
Yeah. And all right, so let me see. Yeah, can you talk a little bit about what animal models and experimental methods you use? Because so you've got this brain region, it's at the bottom of the brain, it's kind of, actually it's right about here.
Yeah.
Right, in fact, you can damage this hypothalamus pituitary if you get
cancer.
And okay, and so how do you study it? How do you know that, say, an AGRP neuron is active under a certain condition, during fasting or whatever?
There was the old school approach, which I think is still used, and it's not unreasonable to use when you analyze post hoc CFOS expression in neurons, that you have a tag. AGRP neurons, which we all knew, or MPY neurons, is the same neurons we knew from the beginning, before the discovery of leptin and these pathways, that you cannot visualize them with an immunostaining, because these are fast-firing inhibitory neurons, so the peptides are so rapidly sent out from the cell body that you actually can do an immunostaining and label these cells. Instead, in the old days, we had to use colchicine that would block external transport, and then you accumulate the peptides. Now, since the mid-90s or late 90s, tools were available so you could tag these cells with the GFAP, green fluorescence protein, or TD tomato, another fluorescent tag. And then if you expose the animal to the food restriction, and then you perfuse the animal, and then you double stain them for CFOS being a early proto-oncogene that is a proxy for neuronal activation. So that was the main tool that people used.
So when the neurons are active, there's calcium influx, and then the calcium very rapidly increases the amount of the CFOS protein.
Correct.
And then you can just take a brain section and use an antibody to the CFOS.
And then you quantify it.
Yeah. That's still a good method. Yeah.
I think that's still a good thing. And now you have this new methods of putting calcium dyes into the cells again, cell selectively, and then monitor this calcium.
Yeah, that allows you to look in the same cells over time in theory.
In vivo, it's an in vivo approach. So
that's technically hard, right? Because you've got to put an optic fiber.
You do have to put an optic fiber, and
the hypothalamus is really hard to get it.
Correct. But these are the methods that most used to address these questions.
So you mentioned that you've mastered electron microscopy methods and immunostaining yourself. So this just kind of general people who aren't scientists. So if you get to be a independent investigator and get funding, you can hire postdocs.
Yes.
Or you can have graduate students who also, by the way, if there's some young people here listening that are thinking about going into science, if you get into graduate school in the sciences, you actually get a stipend.
Correct.
You don't undergraduate, you have to pay. Graduate in science,
medical school you have to pay too.
Medical school you have to pay, but if you're getting a PhD or even master's, you get paid.
Correct. Because
the money's ultimately coming mostly from the government.
Correct.
From taxpayers' money. But anyway, so you've hired postdocs or graduate that have expertise that you didn't have yourself. This is
so the way I developed this is that I developed hubs of technical expertise, some of which was run by technicians. So electron microscopy, I had an electron microscopist who was a technician. And actually, most of the electron microscopy is done by technicians. Then I had a person who was expert, a PhD of electrophysiology. So he developed an electrophysiology hub, and the postdocs, graduate students could come and learn the technology. I developed people who were very good at behavior. So there was a core. So I developed my own lab using my own course.
Yeah.
Instead of leaving a graduate student on its own which originally I was left alone, so I kind of figured to master a technique. And by now we do understand that a single technique does not really give you a full answer, but introducing them to the various technologies and giving them a vision how you can be aware of what's needed to be done. I think that's the way I developed my lab.
Okay. Yeah, I guess I had it that way too, particularly when I was at NIA. We have kind of permanent positions for people with different expertise. Okay, so let's get back to research. So when you eat a meal, glucose goes up. If you fast a long time, glucose levels stay low, fatty acids are released, then some of them are converted to ketones. And you've done some really interesting work on looking at, you have this model, actually, I've got it up here, where when you eat and glucose levels go up, it essentially, when glucose goes up in cells, it increases free radical production.
Correct.
Something we call reactive oxygen species, oxygen radical. And you have this interesting story where these through these reactive oxygen species, the glucose is affecting the neurons in the hypothalamus. Can you talk about that?
So yes. So it was interesting when we started to look at how HGRP neurons, the hunger promoting neurons, are activated, and how the neurons that promote satiety activated. There are very different circumstances. We found that hunger promoting neurons are activated by long chain fatty acids. And the way long chain fatty acids are utilized within the cell through the mitochondria is a very different process than the way glucose byproducts are utilized eventually in the mitochondria. And just to make that long story short, in our view, and we very strongly believe in that, based on data, of course, long chain fatty acids enable something that is a specific mechanism in the mitochondria to throw protons through the intermembrane space, so that you have less likelihood of an electron becoming a free radical, because the protons are provided, and there you do hydrogen peroxide and you can move forward. It's very different, and it is the uncoupling protein component of the system. Uncoupling proteins are important for this enhancement of proton transport into the matrix of the mitochondria.
And do ketones do the same thing or not?
Ketones might do the same thing on their own. Yes.
Yeah. Okay.
Now, when you have a glucose byproduct, then the transport of the protein, the proton is entirely hinging on ATP synthase, which is an enzyme. Yeah. And the ATP synthase has a limited amount of capacity, so protons can accumulate between the intermembrane space. Electrons can then be generated because they're separated during the process of the mitochondria, and there you have the option to generate a lot of free radicals. While that is seemingly a problem and can be a problem, especially if you continuously elevate satiety, which I'll come back to if you are interested in, this reactive oxygen piece is actually very, very important for the activation of these cells. So in that moment of when you reach satiety, these species are very high, and sort of also sensical, because this is a byproduct of the fuel that you took in. Yeah. Sort of a readout. You had enough of this coming in. Here is the ROS that tells you you don't need more of this, because potentially that can be harmful. So the way we look at ROS is that, as with everything else in biology, it has its purpose, and if it's not in a proper way, then it has its negative impacts on cellular integrity, and of course then through that tissue and organismal integrity.
So the mitochondria, so of course neurons have very complex morphologies, axons, dendrites, and they have to have ATP out in all these processes, and presumably that's coming from mitochondria localized in these processes. You've been studying this particular uncoupling protein that you meant causes like proton leak. And so is there any evidence that this is playing a role in synaptic plasticity in the actual, say, release of neurotransmitter or?
First, I always have to say that we believe so. You know, I'm not in possession of the absolute truth, but based on the works that we have done, we clearly believe that changing mitochondrial dynamics and function, they have an impact on synaptogenesis and the removal of synapses, and also that they potentially have, especially if you look at an exon terminal, where you have potentially large amount of mitochondria, that considering that these changes in mitochondrial function has a huge impact on calcium milieu, on pH, potentially temperature, that they have to have, in our view, an impact on neurotransmission, the transmission of classical neurotransmitters or neuropeptides, which release mechanisms we understand less.
And you've also, in addition to studying, or maybe complementary to studying these circuits controlling hunger, satiety, energy metabolism, you've done some work that's relevant to neurological disorders, right? And so these uncoupling proteins, we did some work with a drug that causes probably much more profound uncoupling than this endogenous uncoupling protein you've been looking at, chemicals called DNP. And so my take on this actually is that this uncoupling is kind of imposing a mild stress response in the cells, and the cells are responding adaptively by upregulating, for example, genes that encode, say, antioxidant enzymes or other proteins involved in resisting stress.
So that basically kind of the preconditioning approach.
Yeah, precondition. Yeah.
I think that's sensical. In fact, I think we did 20 some years ago propose that in association with epilepsy, how that might be beneficial in epilepsy, and then also in cardiomyocytes, how it could be actually beneficial. Yeah. Small little strokes can actually prevent you from a larger stroke. It's almost like pre-pulse inhibition. You have a small events that prepares you for the larger events.
What doesn't kill you makes you stronger.
What doesn't kill you makes you stronger. There is a conceptual angle to it that I usually use as well, and that is that most of these conditions are late onset disorders.
Yeah.
And when you think about late onset, then time becomes a player in these. And we simplify time, probably arrogantly and wrongly, to cellular metabolism. So how you utilize fuels, what are the controllers of the fuel utilization? So time can be different for one person and the other, and obviously people get sick earlier than later. People don't get sick. Some people live until 100. Some people die at the age of 25. So it's a very interesting question that we are trying to pursue, but it's not an easy question to ask, and definitely not an easy question to answer in an environment where your money is hinging on study sections.
Yeah. Funding is always spend a lot of time writing grant proposals, and then a lot of the institutes now, the funding level it's a lot lower than it used to be back in the late 80s, early 90s, when I first got my first faculty position. Funding levels were like close to 30% of the proposals, and now most institutes it's less than 10%.
Correct.
Right, and it's tough.
And what's also tough in this environment, to be honest with you, and probably you experience that too, if you think out of the box, which I think many of us do, otherwise, if people wouldn't think out of the box, things would have been already solved. I mean, if that would work. But in the same time, it's very difficult to convince the environment that a new idea is worthy of support. And there is obvious the reasonable skepticism about them.
Yeah. There's always this fine line between how much preliminary data do you have, and on the one hand, and well, you've already solved your problem on the other hand.
Exactly. That, and it happened, both of them happened. Or another example, which is my favorite, is that it's identical grant of mine would get triaged in one study section and got accepted in another one. So it's an interesting enterprise for sure.
Yeah. One more thing on mitochondria before we move on is so we did some work with a protein called sirtuin-3. SIRT3, it's a protein that is what it does is it removes what are called acyl groups or acyl groups from lysine, the amino acid lysine on proteins. And its function is kind of similar in a way to a phosphatase.
Mm-hmm.
So there have been multiple Nobel Prizes given for work on protein phosphorylation, where phosphate is either added to a protein by a kinase or removed by a phosphatase. Right. In neuroscience field, Paul Gringard comes to mind. Right. Actually, he was there, Yale, for a while.
Yeah.
Yeah.
But
anyway, more recently, this other protein modification by enzymes has been described, acetylation, where acetyl group is either added by acetyltransferases or removed by deacetylases. So anyway, the SIRT3 is only in mitochondria, but we did some work in SIRT3 knockout mice and have some really interesting, we don't know the mechanism, but somehow the SIRT3 in mitochondria is affecting synaptic plasticity. We never really figured out exactly how that's working, but maybe something related to the way the uncoupling proteins do, for all we know.
And when you did that, did you follow the calcium milieu of the mitochondria and how they interacted with ER? And
we did work in cultured neurons that lack SIRT3 compared to neurons that have SIRT3, and we expose them to glutamate. Right, it's kind of the excitatory transmitter. Causes calcium influx, and so in neurons where mitochondria lack SIRT3, they're much more vulnerable to excitotoxicity and much more prone to calcium overload when they're stimulated. So the SIRT3 is somehow in the mitochondria kind of playing an important role in regulation of responses to neurotransmitters.
You look at fission and fusion events under those conditions of the mitochondria.
We didn't. We've done work looking at mitochondrial biogenesis, but not related to SIRT3. But yeah, why don't you describe this? So I mentioned that neurons have a lot of mitochondria and they've got to be distributed all over. Well, when the neuron is born during development, it's a sphere, essentially.
Yes.
Right?
Correct.
And it doesn't have many mitochondria.
Correct.
And then as it grows, you get more and more mitochondria, and some of them move out. So that's mitochondrial biogenesis. But individual mitochondria can divide or they can fuse. And I know you've done some work on this.
Yes. In fact, I have to tell you, not to be a contrarian here about that word.
Oh,
but
okay.
But I see fission as a way of getting more mitochondria.
Right.
So for me, mitochondrial biogenesis and fission has something in common. And I believe that, or we believe that some of the regulatory principles of these events relate to fuel availability and what is available for that cell what types of fats, what types of carbohydrate, what types of amino acids, and that those might be the regulatory components with obviously some proteins that are involved, fission and fusion and so forth. But it is the fuel that determines what's going to happen to that mitochondria.
In our work on intermittent fasting, we kind of develop, and this applies to exercise too, where kind of an optimal cell function and plasticity is promoted by intermittent challenge recovery cycles.
Yeah.
So in the case of bioenergetics, the challenge would be fasting or food deprivation. Yeah. And then the recovery would be eating. In the case of exercise, the challenge is the exercise. It's a stress exercise. Then you need this recovery period. And so we think during the stress period, there's gene expression patterns that are set in motion that then end up doing good things in the recovery. So probably during the stress period, you may not get the fission or biogenesis. That may occur when you've got the nutrients available. Yeah.
And something that for me conceptually fits in the same thing is circadian rhythm, because that's sort of the same push-pull kind of thing. You go in this direction for a while and then you switch to the other direction in a while. So these rhythmic things are intriguing to me in general.
We have a granddaughter that's 6 months old now, and
congratulations.
Yeah, so that reminds me of her. So this is well known. So these developing kids, when they grow more when they're sleeping than when they're awake, right? So when they're sleeping, it's kind of a recovery period from they're crying or whatever.
Yeah. And that's cool. I didn't think about it that way along those lines.
Yeah. So the more and growth hormone goes up when you're sleeping.
And in fact, during that period, their wakefulness is predominantly because they are hungry and they want to eat.
That's true. They're always hungry. Yeah.
So it's kind of
my daughter-in-law is still breastfeeding, and it's yeah. Of course, they're hungry more frequently because they're growing very rapidly, right? Yeah.
Interesting questions.
Yeah, I don't know how much this hypothalamic system has been looked at. It's I guess technically it's harder to do in, say, newborn animal than an adult. That's because everything's smaller. But you could still do like the CFOS stuff and do some manipulations or something.
Yeah, the problem in part with the animal models of postnatal period is that it's really not the same in humans. It's
that's true.
And that I think gives a little bit of a bias towards what they start to move early. They go, and by the 21 days of age, they're kind of, I'm not saying young adults, but definitely can independently feed.
Yeah.
But maybe you can find a early moment. One of our colleagues at Yale, who used to be working with me and now he's a faculty, he's studying these kind of transition, what these neurons do in the early development versus what these neurons do in the adult, and seems to be significantly different in the early period, more involved with social interactions with the mother, and eventually they switch into interaction with the food and environment more.
Wow. Oh, that's really interesting.
It is.
Yeah. Let's see. You haven't talked about anorexia. You've done some, anorexia nervosa, psychiatric disorder. It's still a problem, particularly in adolescent girls. Seem to be most affected. And one hindrance to really understanding, what's happening in this human condition, that it wasn't, rats or mice don't normally starve themselves, right? I mean, it's not. So you've developed a model of anorexia that now enables you and other people to try to figure out what's going on. Can you talk about that?
Yeah. So actually the model was developed by others, but we exploited it for the question of to what extent these AGRP neurons, the hunger promoting neurons, might be relevant for the disease, the etiology, and the outcome of the disease. You know, when you think about anorexia nervosa, I think thinking about the hunger promoting neurons is reasonable because these individuals self-suppress their eating despite of the fact that actually they're hungry. And they frequently over-exercise. It's one of the outcomes of that. And all these events, the starting point frequently, if not almost exclusively, in the peri-adolescent period, that there is also a large social stress that impacts these predominantly women. So what we did in order to ask we also asked a very simple question: to what extent, again, AGRP neurons might be relevant for phenotypes of anorexia nervosa? And what we did, we put animals in cages with running wheel. Ad libitum food was available, and mice like to run on the run. But then we restricted their availability of food in the dark phase, their active phase, for 2 hours, a limited amount of food. And what became obvious is that they really started to be very addictively run on the wheel. They did eat their food, sometimes not all of it, but in the absence of, once the food was gone, they were running and they were running quite a lot. Two things what we found, which are sort of seemingly counterintuitive, but I think speaks to the disorder. One of the problems of anorexia nervosa is that this is has the highest mortality among psychiatric conditions other than suicide. And the answer to that still is not known, but I think our study shed some light on potential mechanisms for that. So when we decreased the activity of AGRP neurons, so they were less available, the circuitry was disrupted, these animals still jumped on the wheel and started to run, but they all died in the next 2 days. Versus when we activated the AGRP neurons during the same period, they ran much better, and their endurance exercise was much better than the control animals, and none of them died. So 2 things. One, it seems that activation of the AGRP neurons propagates compulsive or stereotypic running, and endurance exercise is actually promoted by AGRP neurons. And the other one is that the absence of AGRP functionality, the animals, if they are not food restricted, they don't die, but if they're food restricted, they die. What we found was that the cause of that was that AGRP neurons are also fundamentally important for your mobilization of stored fat during the time of hunger. You need alternative fuel, you get it from the fat. And they were incapable of mobilizing food. They were incapable of propagating proper gluconeogenesis. So all the fuel pipes dropped to 0. And they died of, we think they died of ketosis, because the ketones were actually elevated in these animals. And they were just indiscriminately burning, and they had not the capacity to stop running. The running was so addictive for these animals that they chose running than life, if you want to put it that way. Now, when you activate AGRP neurons, they run more, and actually their profiles were very good. When you stop the paradigm, and eventually you see that the control animals retain certain anxiety phenotypes, learning and memory phenotypes. If you have elevated AGRP neurons, they recovered from those things. So in essence, the anorexia nervosa model that we used suggests that yes, AGRP neurons might be important in the etiology of propagation of these, eating less, moving more, but they actually do it in a way that the animals survive, versus if you don't, then they don't. And then, although we didn't put in the paper, but I think what makes us think about it is that actually it could have been at certain times a positive trait. When you need to migrate out from a space where you don't have food and you don't know where is the next spot, having this adaptive response could be actually beneficial, because you may be able to move despite of food and eventually find your food source. So
yeah, since
there's
there's
a lot of interesting,
ideas that from an evolutionary perspective, if there's no food where you're at, you better get moving.
Moving and move, and hopefully you'll find it.
But then somehow, the humans with anorexia nervosa, they do have food available. It's just that they purposely don't want to eat it.
Correct.
So that may be just an additional psychological thing that, social body image.
Absolutely. Body image is a critical element in this, and frequently preceding the engagement is this, they tend to be a little bit on a heavier side. So there is that social stigma, bullying, and so forth that have an impact. And there is another aspect, I believe, that if you cannot control your environment, you can control your own body. That you can. So that's an interest, raises a lot of questions that
that's something they can control. And,
yes. Yeah. Correct.
Yeah, interesting. Okay, let's go back to obesity. You briefly mentioned the GLP-1 receptor agonist, the Ozempic. I don't know the trade names so well as their actual names. So exenatide,
Mounjaro, which is a dual agonist with the GLP.
Yeah. Now the receptors for these. So you mentioned that these GLP-1 receptor agonists can cause nausea. So they inhibit food intake, and probably that's a big factor in their mechanism of action. But their receptors they're widely expressed. Are they in, I assume they're in AGRP and POMC neurons?
There are studies that you can find that they have it, and you said, but I think the emerging data is that it's going to be acting through the vagal afferents and through that pathway towards the parabrachial nucleus, which kind of makes sense. And actually, they don't really have to enter the brain this way, because they're hanging out, on the vagal afferents. I think that's the direction.
So there's been experiments where they say you have a mouse or rat obesity model, right? Diet-induced obesity, or there's mutations, say, in leptin receptor, and they overeat. Anyway, so you have mice or rats that are obese. If you then give them the GLP-1 receptor agonist, they'll lose weight. But what happens and so did they experiments been done where they cut the vagus down low somewhere and show that they don't respond to the drug?
I know that many papers are coming out now because it's a very popular thing. I believe that there are some older studies from 10, 15 years ago where they did similar studies. And I believe there is a consensus that that's how it goes. But I'm not an expert on this, but I wouldn't be surprised if that's going to be the outcome of this thing.
It kind of makes sense. So the GLP-1 is in these specialized epithelial cells in the intestine. It's released into the blood mainly in response to glucose, I think. And and then so these vagal nerves, their endings are innervating around the gut. So probably the highest concentration of GLP-1, these vagal afferents, that is the fibers that convey the information from the gut area up to the brainstem, they're right there. So maybe that makes sense.
I think that makes the easiest sense. Yeah, I think that's sort of sensical.
Yeah.
But we'll see how it's gonna work out. Again, we'll see.
I still don't like the idea of you know, this may discourage people to exercise and to quit eating junk food and overeating.
So I gave a talk I gave 2 talks in Korea. One was actually on a obesity therapeutics meeting, and we published the paper with my postdoc back in 2012 on our concerns about obesity drug development, where you promote satiety, which we based on our cellular observations, as the ROS issue that I talked to you about, that satiety, you are promoting ROS. You can go around that. And that if you you can succeed, but the long-term consequence is gonna be you won't know until 20, 30 years. Yeah. So when they say, oh, they've been in the market for 10, 15, I don't know how many years. First of all, these large doses are not. These are new because they are dosing them up for have the proper weight loss. We shall see. And I agree with you. I think, and I know it's sort of common sense and boring, but behavior adaptation or modification is the best to deal with these issues.
And presumably then we mentioned this stress recovery. So in this case, if you take the drug, you're not exposed to the stresses associated with hunger and the evolutionarily ancient pathways that evolved to adapt to these. So yeah, we'll see what happens.
We'll see what happens. Exactly.
Okay. Well, we covered a lot. And what's your most exciting studies going on now, without giving away the...
Well, we have been interested in the last couple of years about the role of these metabolic signals or hypothalamus in schizophrenia, both from the perspective of development as well as adult, propagation of symptoms. And it comes from very, very simple original ideas. Insulin-induced coma was used very successfully to treat positive symptoms of schizophrenia up until the 50s. Of course, there were deaths, so eventually, they stopped that. But then you look at the other side of the coin, the psychotropic drugs, clozapine, that are successful in treating the patient's positive symptoms, induce massive obesity and diabetes, arguing that there is a fundamental problem with glucose utilization or metabolic principles of certain pathways of the brain. We don't dispute that there are genetics or epigenetics factors that make certain pathways in the brain vulnerable, so that you could almost say that they flip-flop between wakefulness and sleep. Because if you think about delusions, that's fine when you are dreaming. It's okay. But the problem is when you have that delusion when you're awake. So we agree that there are genetics in it, but we think that there is a metabolic element to that is worthwhile to pursue.
And these antipsychotic drugs, they mainly affect dopamine systems. Is that, do you think there's a connection then with the hypothalamus between the reward, the dopamine system?
A paper we published also 2012 shows that if we, again, interfere with the AGRP neurons, we end up with a very different midbrain dopamine system, both in its way to respond to signals and both its projection side. So definitely altering these metabolic regions of the brain can have broad impact on brain development. And this has happened during, let's say the second trimester of pregnancy. So definitely we believe that at least they have a role to play. And we argue for that because there's not much that has been accomplished in the understanding of these disorders, and maybe coming from beneath is a way to ask the question in different ways.
Yeah. Interesting. Who did I have on? Oh, Danny Weinberger. I had Danny Weinberger on. He's at Hopkins. He has this Lieber Institute. He's done a lot of work on the genetics of schizophrenia. And there are some animal models, but it's really hard to say.
Very hard.
You know, because you can't. Is the animal having hallucinations? I don't know. I mean, how do you know that?
In fact, at 2:30 this afternoon, I have my graduate student who starts with me and wants to work on that project. And I will have to explain her that it's, with the animals, predominantly what they look at is locomotion. They look at how they enter the center of the open field, the open arm on the. So yes, when maybe a mouse becomes a buddy with a cat, then that's a delusion. You see.
Well, that's interesting. Huh. Okay. Well, I enjoyed seeing you again. It's been quite a while. I had a postdoc in my lab who was from Hungary. Remember, I don't know if you remember Christina, and she did well and she moved on, doing well. What's going on in Hungary now from the standpoint of science and funding? Is it okay? Is this semi-authoritarian government causing trouble, or?
The problem is, first of all, they always had a very archaic funding system from within the country. It's very corrupt in a sense that few people get money, but that was there already a long time ago. And then this new development is that because of the current government, the European Union is not allowing funds from the European Union go to Hungary, which is a big problem.
Why? I know why they're not allowing to go to UK, but why not Hungary?
Because the Hungarian government reorganized the university systems in a way that they find not to be democratic.
But Hungary's part of the EU, but they're not.
Correct. But they're withholding a lot of money because they don't like the government's activities.
Oh, I see. Kind of a sanction, I guess.
Kind of a sanction, yes. So we'll see how it goes.
Yeah, but it's not good to sanction the scientists. They're just trying to do their work and.
Correct. Right? Yeah. That's how it always works.
That's too bad.
Yeah.
Okay. Well, Tomas, I know you're busy. I'm retired now, so I'm not as busy as you. And now, do you oversee all the animal facilities? You have someone else?
No. I in my department there is a head of the entire facility, and he reports to me. But he's the attending veterinarian.
Yeah. So you don't have to mess with the animal care and use committee too much, or?
No.
Yeah.
Good.
When I was at NIA, this one we started doing the intermittent fasting calorie restriction studies. And I we have to have the protocols reviewed, right?
Okay.
And they were concerned I didn't justify, subjecting the animals to this harsh treatment of every other day food deprivation or something. And I turned it around on them and I said, everyone else need to justify why they keep their animals in obesity, no exercise, they overeat.
It's remarkable what you're saying, because when I was running my and still has lifespan studies, we have to put in category E because it's based on them, it's death of the endpoint. I mean, we're giving the entire life to the mouse.
Right. You're just documenting when they die natural death.
Naturally, we give them the entire life instead of killing them, 3 months.
And of course, ultimately you euthanize them with overdose of anesthetic or whatever when they get sick enough.
Correct.
So it's not like you're letting them really suffer a lot.
Yeah, exactly.
Yeah. Okay, well, these are the kinds of things we wrestle with.
Yes, such is life. Thank you very much, Mark. I really appreciate it.
Yeah.
Good. Keep in touch.
All right. Bye.
Bye-bye.